Welcome to the AME Food Testing Show. Today's topic, Leatherhead opens DirtyLab for pathogen challenge testing with John Haines, Principal Scientist, Food Safety, Leatherhead Food Research. John has 23 years experience working in clinical and food sectors, with 16 years experience and service at Leatherhead, working on development, optimization, and evaluation of new technologies for the rapid separation, concentration, and detection of analytes of importance to food safety and quality, including foodborne pathogens, toxins, viruses, allergens, and adulterated ingredients. Technologies for separation and concentration include SPE, immunoaffinity, magnetic separation, dielectrophoresis, free-flow electrophoresis, and molecular imprinting. And for detection include real-time polymerase chain reaction, immunoassay, microarray, optical biosensors, and fluorescence microscopy. Current work includes the development of methods for the detection of antibiotic-resistant bacteria, ESBLs, and virus, HNOV, and surrogates, inactivation during food processing. Leatherhead Food Research delivers integrated scientific expertise and international regulatory advice to global food, drink, and regulated industries. Now, welcome with me, John Haines, Principal Scientist. John?
Yes, hello. Yes,
welcome to the show.
Thank you very much. Just to summarize your introduction there, it's pretty up to date what you said. We do specialize within food safety at Leatherhead Food Research on looking at new methods for detection, but obviously within the food sector, it's often the sample preparation stage involving separation, concentration forms a critical part of that. So those sample workup methods linked with the methods of detection are an important part for us. And what we're currently working on at the moment is a range of projects. Again, as you briefly mentioned, we are looking at a project looking at antibiotic resistance, and for that we're using a mixture of sort of traditional chromogenic selective agars, but also linked with a LAMP assay. It's a molecular assay. And on viruses, we're doing a lot of work on human noroviruses at the moment, looking at inactivation. And as part of that, we're doing some modeling with certain parts of the food processing setup, and that we're transferring to our new DirtyLab facility. So hopefully that gives you an indication of what we're up to here at Leatherhead.
Well, John, thank you so much. Just for our information, your lab is located in the United Kingdom. Let's begin with a very general question. What is a human pathogen?
Well, in its widest sense, a human pathogen is a microorganism, if you want to call it that. It's like a bacteria, a virus, a fungus, a mold or yeast that causes disease in humans. So obviously, it's important that steps are taken within the food manufacturing and also in the retail and the consumer part to identify the risks and try and minimize the risks as much as possible throughout the whole of the food chain.
John, how do these pathogens contaminate food products?
Well, contamination really can be introduced at any point in the food chain. So right from the raw ingredients coming into the factory or being introduced into the food processing stage, and then throughout food processing, you may get contamination from different foods, different ingredients. Also, there's always a human element as well, sort of sanitation or the cleanliness of the procedures in place, and contamination possibly coming from the people working on the processing line. So again, this can introduce things like staphylococcus and indeed norovirus, for example. But then it doesn't stop there. It doesn't matter how good the manufacturing process is within factory. Once it leaves the factory, there's still scope for contamination to occur. So it's through the distribution network at retail, and obviously when you get it home as well. So there's a risk of contamination throughout the whole chain. So again, although we talk about risk prevention and the introduction of hazard procedures within the food manufacturing process, this whole idea of risk, identifying the risk and controlling that really does follow all the way through to the end, the end consumer at home.
Well, John, as you have established a DirtyLab, what are the methods that you'll be employing to test for these pathogens?
We do, even before DirtyLab, I mean, at Leatherhead, we've had a long history of carrying out challenge studies and shelf life studies. So, again, in the traditional challenge study, you're inoculating your particular food or ingredients with known pathogens and seeing how they, basically how they survive within that particular product under certain conditions. So what we're going to do, the idea behind the DirtyLab, is really to be able to, it really is a question of scale, really. What we're trying to do is model the system to nearer what actually goes on within the food manufacturing process at factory. Because we've done a lot of work over the years on looking at different inactivations, heat inactivation, heat and pressure inactivation, disinfectants and so forth, UV, whatever it may be. And whatever you get in a relatively small scale on the bench top in the lab, it doesn't always transfer through to when you scale up to the sort of volumes or sample quantities you see in the factory. And this, the physical parameters of heat transfer, whatever it may be, on surface or through mixing, for example. It's a combination of both the physical, but also basically the equipment you're using. That plays a massive part in how for example, if you're eliminating microbial contamination within your sample, the equipment you're using does play an important part. So really, the nearer you can get, although you may still be modelling to a certain extent, the nearer you can get to sample size, sample scale within the food manufacturing process, the closer you are to really defining the real risk to your particular product.
Well, John, Leatherhead's new pathogen pilot plant, as you said, is a DirtyLab. Can you describe the mission in general terms?
Well, yes. I mean, the basis behind it is the ability or to have a setup where we can validate the effectiveness of different methods, processes, and equipment, manufacturing equipment, against known microorganisms, including obviously the major pathogens. And this doesn't just look at challenge testing in its conventional state, but also looking at not only the effect on the food products, but also on the food processing part, the equipment, and also the cleaning and disinfection of that equipment. So within the DirtyLab, we can effectively bring in pilot scale or prototype equipment, contaminate that under containment level 2 environment, and both test the food product and also the equipment. And as well as that, we can then at the end we can look at the disinfection processes. And again, this all links in because different foods on different surfaces may well form biofilms, for example, and this plays directly into how well the cleaning or disinfection procedure works. So this is another good reason why the nearer you can get to a really good model, as near as you can get it to production scale, and using the same equipment, the same materials, the same surfaces, that really does gives you a good idea on how the product and how the microbial component within that operates under those conditions. So you have set conditions of temperature, humidity, gas, light, and pressure, for example, if you're modelling the extrusion process. And these are all important parts to try and model as accurately as possible to really give you the overall picture of what's happening to your product.
Well, John, that's certainly a fascinating perspective and a very helpful, I'm sure, data set for your clients. I'm curious, which processes do you use to deliberately contaminate the food products with pathogens?
In terms of, if you're talking about deliberate contamination, you put your finger on it. That's difficult to model because you don't, within the process, you don't tend to get deliberate contamination. I say what you would get was unexpected contamination through either like cross-contamination from product to product. So it may be as part of your process line, you may have raw meat, for example, or eggs or whatever, which may contaminate another process line. You may have someone not following, for example, the hygiene procedures correctly, and you get through physical handling of either the product or one part of the processing equipment, you may get contamination from the human element. So basically, that's what we're trying to model. But in terms of how we would contaminate it within the DirtyLab, obviously we would actually identify what are the critical control points. Again, following the standard sort of HACCP procedures, we would know, again, in discussion with the customer we're working with, we would identify what their critical control points are within their HACCP program, and sort of contaminate at those points and see how the process reacts to that, and how, for example, their cleaning procedures, how well they work to get rid of any contamination that may be present.
Well, John, that data that you are providing in this challenge testing in a controlled environment, which exactly replicates your client's production areas, how does that data help your client?
It's really it basically gives them confidence. What they've got is they've got sort of best practice guidelines which they've put together again over time, and this may be based on some published data, as well as some experimental work that they've either carried out themselves or they've had produced for them through a research laboratory. But as I said before, if that's being produced in a laboratory on a relatively small scale, it gives them an indication of how it's going to work with their product and in their process, but it doesn't give them the full picture. So again, if we can model their system as near as we can get it to the final production process, basically the more confidence they will have in their limits. I mean, obviously, as part of the food safety, they will still be carrying out their regular screening, so auditing incoming raw ingredients, doing final product testing, for example. But as part of the general HACCP procedure, if the systems they used and the data that's been generated on a really robust model, and I think that's really the critical thing here is. It's robust data based on a really good model of their system. It just gives them more confidence in the statements there, which they're able to, or the claims they can make on the safety of their process.
Well, John, if you could, how would you describe your optimal client and how you could help that client, as we talk in general terms?
That's quite a difficult one, because the way Leatherhead operates, we are as an independent company. We've got over 1,500 members worldwide, so we are sort of every organisation. But basically, within that, we cover all aspects of the food industry. So it's raw ingredient suppliers, it's food manufacturers, it's distributors, it's retailers. We have input from consumer groups, consumer organisations as well. So we do work, and not just necessarily intended work for DirtyLab, but across the whole company. We do work for all parts of the food sector. So again, what we try and do is obviously tailor work for those individual sectors. But what we would hopefully to do with the DirtyLab, it will primarily be for food manufacturers. So as an example, what we're doing at the moment is modeling the extrusion process, which a particular company uses as part of their food manufacturing process. And again, this particular company has it's a multinational, has many different manufacturing sites, and you know that by taking data from different sites and use of different extruders, there is quite a variation in performance. So again, if we can get some realistic model of the extrusion process, and so for that, we're looking at the combination of heat and pressure on the inactivation of pathogens within their particular matrix. So the way we work with companies is in close discussion with them. We work directly on their particular product. So it's that's exactly what the final recipe of the ingredient list is. Well, that's what we're using. And then we take from them their industrial parameters, and we model directly on that. So in terms of the extrusion, and the extrusion process, as an example, is quite hard to model, but we have got a setup now where we have a pressure reactor system, which we can use to model and basically look at the inactivation of pathogens under certain temperatures and certain pressures. So that's the way we go about it. So really, it's really the methodology we use is worked up to fit the requirements of individual customers.
Well, John, I see a big distinction between your sample laboratory, which models your clients in a separate location than the actual production, as opposed to a third-party lab receiving samples and then sending the results back to the client, detached from the actual process. So you're duplicating the process. And I was wondering if you could conclude our discussion with a brief review of what is a human pathogen, how do they contaminate foods, what are the methods that you have employed at your new DirtyLab, and what benefit you bring to the industry to improve best practices.
Yes, certainly. Well, I mean, as we discussed, I mean, a human pathogen, and in our work, we're mainly looking at foodborne pathogens, things like Salmonella, Listeria, Campylobacter, botulinum, as well as viruses, things like human norovirus, rotavirus, for example, as well as molds and yeast. So we're looking at what processes work best under a typical manufacturing procedure to eliminate or minimize the risk of getting contamination from these organisms within a particular product. So we're looking at, and as you rightly said about sort of third-party testing, obviously we still employ the standard test to determine the presence or absence of these organisms. So we generally use RT-PCR for detection, also in relation to the standard cultural method. So we have basically two results to go on, and really again the idea and the overriding idea of this is to get the most robust model in place to clearly show basically the risk potential. So not just the benefits, but also what's going wrong in certain cases with the working practices that the customer setup is actually using. And as an example of that, we have observed that different equipment will have slightly different materials, different whether it's sort of a smooth finish or a rough finish in terms of the stainless steel internal lining. I mean, this, for example, can play directly into how well pathogens attach to a surface. So again, if they attach, they can form biofilms, and that gives them certain protection against the disinfection and cleaning procedures. So It's really important to try and, as I said before, to model the system as closely as possible to the manufacturing process, so you get the most in-depth understanding of what the risks are. And again, it may well show that the procedures you're employing work perfectly well, which is a great result. But if it flags up certain areas, and it may be at certain points along the processing line where you may have problems, for example, if your cleaning procedure is not cleaning in a particularly productive way in certain points, then you know that you can go back to that particular part and work up better methods to clean your procedure. So, I think as an overall conclusion, it's not. There's nothing particularly new in terms of the overall science, how people carry out challenge studies, how they assess risk, and how that's then integrated into a hazard procedure. But I think the big thing about what we're trying to do with DirtyLab is it's really a question of scale. It's trying to scale up to a working model that is a robust model for the manufacturing procedure, and this will give us really the best understanding that we can possibly get for basically how the manufacturing process works, and how and what the risk is in terms of contamination, and the ability of your cleaning procedures to remove any contamination.
Well, John, you've certainly given us an interesting perspective into the mission of the DirtyLab, and I see it a combination of the effectiveness of good manufacturing practices, environmental testing, and product testing, all in a real-world simulation that could offer real troubleshooting and optimization data to your clients. Would you like to offer any final comments before we close our interview?
Well, yes. Your summing up there was totally agree with that. That is basically what we're trying to do, and again, it does tie into what companies perceive at the moment as best practice, and just trying to give them a way of actually producing data to support their beliefs, because when people put together, some of it is taken on trust to a certain extent, that if you've shown a particular inactivation process works at a relatively small scale in the lab, that it will therefore work once you scaled it up 100 times in the factory. It's really essential to get some sort of validation data on that larger scale work to actually show that what you've shown in the lab does actually transfer to the larger scale. And I think that's important across the board. It's all to do with getting confidence basically in the data, and so that when you're putting claims forward in support of your GMP and best practice guidelines and so forth, you have got the best data to support your claim.
Well, John, I'd like to thank you for your participation today and wish you a great day. If you have any new developments that you'd like to report back to our now 60,000 listeners who are primarily food production, food quality, food safety, and food security managers, feel free to contact me and get back on the show.
Well, thank you very much, and it was very good to talk to you. I'll certainly like to keep in touch, to see what, again, having a look at some of your other interviews. It's very interesting stuff that you're doing. So, yes, I'd certainly like to keep in touch.
Thank you very much. Have a great day.
Yeah, and you.
Bye now.